Please join me in welcoming our next speaker, Dr. Monica Arienzo. Dr. Arienzo is an assistant research professor in the Division of Hydrologic Sciences at the Desert Research Institute. After she got her bachelor's degree in geology from the Franklin and Marshall College, she earned a PhD in marine geology and geophysics from the University of Miami. In her research, Monica uses chemical tools to understand how humans have impacted the environment. After years of studying caves in the Bahamas and ice cores from Antarctica, now she studies pollutants and the climate change impacts in the Western United States. Her current research focuses on pollutants, including microplastics in snowy peaks, downstream lakes and rivers, and drinking water taps. Today, Monica is going to talk about microplastics in the Sierra Nevada, from peak to pipe. Welcome, Monica, and I now turn the floor over to you.
Excellent. Thank you so much for that wonderful introduction, and thank you all for joining us today. Today I'm going to be talking about work we've been doing in the Sierra Nevada to study microplastics in this environment. You can see in the background of my slide the snowy peaks of the Sierras, and in the foreground you can see here Lake Tahoe. In the wintertime, snow accumulates in the Sierras, and then the spring and summer that snow melts, and then the water then moves from the snowpack down to the downstream lakes and rivers like Lake Tahoe. And so my interest is understanding how microplastics are moving in this environment. I'd like to thank my collaborators on this project who have helped us to collect the data and develop these methods that I'll be talking about today. As we're all really busy these days, I wanted to start with my takeaway points. And so what I'll be telling you today is that the combination of stainless steel filters and peristaltic pumping method enables sample flexibility and streamlined laboratory processing for microplastics. Stainless steel filters are applicable to micro-FTIR for microplastic detection in samples. And lastly, I'll be showing you that a variety of different microplastic types were identified in Lake Tahoe surface water. And so the outline of my talk today, I'm going to begin with an introduction to the study. I'm then going to introduce you to the peristaltic pump and stainless steel filter method. We'll then move on to talk about the application of those filters to micro-FTIR. And then we'll end by talking about the application of these methods to Lake Tahoe. All right, let's begin. So what are microplastics? Microplastics are typically defined as particles less than 5 millimeters but bigger than 1 micron. And so this is about the size of a pencil eraser down to the size that's a little smaller than a bacteria. Here in Amanda's hand, you can see some micro and macro plastics from plastics that she collected along the beach in Lake Tahoe. So why are we interested in studying microplastics? Well, there's a lot we still don't know about microplastics. And as a geochemist, my interests are really in trying to understand human impacts to the remote environments. And so I've been spending a lot of my time thinking about impacts to places like Antarctica, but I wanted to bring my work a little more locally. And so some of the questions we have about microplastics are things like, what are the sources of microplastics to the remote mountain environments? Does the concentration of microplastics vary spatially or temporally? And what are the potential impacts to downstream water resources? And so I'm really lucky, right nearby where I live here in Reno, Nevada, the Sierra Nevadas are a perfect laboratory to study these questions. For those of you who are not familiar with the Sierra Nevada, this is the mountain range located in eastern California, close to the border with Nevada. And so these mountain ranges are a nice laboratory to study these questions because we have remote areas, so we can study sort of the pristine nature. But then there's also areas that humans frequently visit and travel to. So we can look at these variations in areas where humans visit and potentially are impacting, but then also areas that are remote. Additionally, a lot of work has been done historically in the Sierras. And so we have a wonderful background of literature that we can add to. All of the data I'll be showing you today are coming from Lake Tahoe, which is located on the border of Nevada and California. When we think about how microplastics are moving through these mountain environments, this graphic is supposed to show how we think they may be moving. So we think, for example, atmospheric deposition may be important. Runoff is probably important for transporting those microplastics. We also have human impacts like the breakdown of larger pieces of plastic to form microplastics, as well as other things like tire wear and tear may also be important. And so this is sort of a general framework that we're working with in thinking about how we're going to build our research to study microplastics in these environments. So in order to study microplastics in the mountain environments, we've been using three main approaches. We've been studying microplastics in the snow, microplastics in the water, and then we've also used a citizen science-based approach to understanding some sources of microplastics. And so here you can see me testing out our citizen science method at a stormwater drainage pipe. But today I'm going to be focusing on our methods for sampling water. And so now I'm going to be telling you about the method we've been developing, which is our peristaltic pump and stainless steel filter method. So when we sample for microplastics in water, there are several different methods we can use, and I'm just going to give a quick overview of some of those methods. One method that's commonly used is the Newston net method, and this involves dragging a large net behind a boat. And this allows you to filter large volumes of water through this net. You then rinse the material that's collected in this net out, and then you bring that material back to the laboratory and you process that there. There can be some drawbacks to this method. It can be difficult to rinse all of the material out of the net, and you wind up with a fairly complex sample matrix using this approach. The other approach that's sort of on the opposite end of the spectrum would be grab sampling. And grab sampling involves collecting samples in bottles. Typically, your volume of sample is much smaller. But you bring those bottles back to the laboratory and process them there. And so you have a lot more flexibility in the filter types you use, as well as you potentially have less matrix because your volume is less. And so then in between these two methods is this ASTM pump method. This uses a submersible pump to then pump water through a series of stainless steel sieves. Through this method, you can filter large volumes of water, but you do need to backflush your sieve in the field if you're visiting another field site. And so there's the potential for contamination or losing some of your sample in the process of backflushing your sieve. And so in studying the literature and learning about these different methods that are being used, we wanted to think about sort of what is it that we were looking for in our sampling approach to address our needs to study these remote environments. And so the list of needs that we came up with is that we wanted a sampling and laboratory approach that would limit contamination and limit sample handling. We wanted a lab and sampling approach that would maximize sample volume. We wanted to streamline the isolation of microplastics. And then we wanted to ensure we could enable the detection of microplastics using micro-FTIR. And so in coming up with these requirements for our sampling and laboratory procedures, the approach we came up with that we thought would meet these needs was this peristaltic pump and stainless steel filter approach. So our peristaltic pump approach pumps water from the body of water that we're interested in through this tubing and then through a series of stainless steel sieves, stainless steel filters that are in line with our filter, or with our peristaltic pump. And you can see a picture down here of what this filter stack looks like in the field. Some of the benefits of this system is that it is portable. So we actually backpacked into some sites to collect our samples. It is adaptable to a variety of sampling locations, water types, and filter sizes, so you have a lot of flexibility. You're reducing contamination potential because your sample is isolated from the environment. And one of the cool things that we also did was we actually would keep our filters in these casings, and we wouldn't open those casings until we were back in the lab. And so that also helps to reduce sample contamination. And this approach also allows us to maximize that sample throughput because we can filter over 100 liters in less than 6 hours. And so we were filtering about 100 liters and able to visit about 2 sites per day. Okay, so once we've collected our sample in the lab, then we have to process it. And so what we do is we keep our filters in those filter casings. We return those filter casings to the laboratory, where we then do a series of steps to isolate microplastics. And so for our samples, this typically includes the removal of sediment and organic material. And there are several approaches in the microplastic literature for ways to remove sediment and organic material. And what I'll be presenting is the approach we have found that works best for our samples. So what we do is once we've returned these filter casings back to the lab, we first want to remove that organic material. And so we add 10% hydrogen peroxide to that filter. And we allow it to incubate in there for 24 hours or however long is necessary for the reaction to occur. So after we've removed our organic material, we then, if we find sediment in our samples, we would then want to remove that sediment material. And so we will back flush that filter using a dense salt solution. And this dense salt solution allows the microplastics to float and the sediment to sink. And so what we can then do is we can then separate those two and filter the supernatant back onto the same filter and remove that sediment from our sample. And so now we've effectively isolated those microplastics and removed these other materials. So the first thing we wanted to do after we developed this method was we wanted to test it and see if we could effectively recover microplastics using this method. So in order to test this, what we did was we counted polyethylene beads. And so you can see these beads here. They're about 125 microns in size. And you can see the background filter here is about 100 micron filter. And so we counted these beads and then we put them through every step of our process. So we counted the beads, we then put them through the peristaltic pumping method, and then we counted them afterwards to see how many beads we could recover. And we found we recovered about 88% of those polyethylene beads. So this is really promising for this peristaltic pumping method in that we're not losing plastics when we are pumping. We then took those beads and we subjected them to each step of our lab process, and we found in total we were able to recover about 71% of the polyethylene beads. And this is similar to what previous studies have found, so this is very promising. I also want to note that the stainless steel filters were resistant to corrosion during these laboratory steps. And we also observed minimal blank when we did a blank test. Okay, so now I've shown you the laboratory methods and the field collection methods. So now let's talk about application to micro FTIR. So micro-FTIR combines Fourier transform infrared spectroscopy and a microscope in order to allow us to identify unknown particles down to a few microns. And so for us, this is a really effective approach for identifying plastic type. So when we look at our samples under the microscope, there's going to be potentially not only in plastics, but there may be some other particles in there. And so we want to be able to identify what's a plastic and what type of plastic is it. And so what we wanted to do, so one of the issues we run into with micro-FTIR is that certain filters can have an absorption limiting IR measurement approaches. And so what happens is that you have to account for that in your measurement approach, or you have to physically pick particles off of your filter and look at them on an IR transparent slide. And so here at DRI, we've been testing these filters, and we've been working with Suja at Thermo as well, who's been testing these filters, and we've been trying to determine how effective these filters are for micro-FTIR. And so in order to test this, what we did was we took those same filters we used in the laboratory testing phase. So you can see here, we still have those 125 micron polyethylene beads on this filter, and we mapped about a 3 millimeter by 3 millimeter area on the filter, and we used transmission mode and the imaging array detector on our micro-FTIR. And so we mapped this area on this filter, and then we used the Omnic Picta software and asked the software what particles were present on this filter. And so as you can see here, the software identified polyethylene, the beads as polyethylene, which is what they are. So this worked. And so this is really promising for being able to use these mesh filters in transmission mode. And so here I've shown you data from a 100 micron mesh filter. We also tested a 20 micron mesh filter using some somewhat smaller polyethylene beads, and we also got very promising results from those as well. Okay, so now I've shown you that we set out at the beginning certain criteria that we wanted to meet in our sampling and lab procedures. And so what I've shown you is that this approach that we've developed for our needs allows us to limit contamination and sample handling. We've been able to maximize sample volume. We've been able to streamline the isolation of microplastics. And we've been also able to detect the microplastics using micro-FTIR. All right, so now let's apply this method in the field. So here we've been studying the microplastics to Lake Tahoe. As a reminder, Lake Tahoe is located on the border of Nevada and California. This is a high alpine oligotrophic lake. We chose this lake to study because it's an important drinking water resource, both for the communities around the lake and communities downstream. For example, this is an important water resource for where I live in Reno, just downstream of Lake Tahoe. We studied 6 different sites in Lake Tahoe. You can see those sites here, and I'll be showing you data from 2 of those sites. And so the data I'll be showing you today are some preliminary observations of plastic type and some pictures of what we observed. And all of these samples were collected using the methods that I outlined earlier. Okay, so what did we find? So we've identified in our filters from Lake Tahoe polypropylene. This is a very common plastic. You can see it's this blue sort of fiber-like particle here. We also identify nylon, and that's this fiber here. We also identified polycarbonate in our samples. These days we see polycarbonate a lot as those sneeze guards that we interact with, and that was this particle here. And we also identified polyethylene in several of our samples. Again, this is a very common plastic that we interact with from making up things like those plastic grocery bags, as well as laundry detergent containers and things like that. And we also identified rubbers in our sample. This is one of the particles we identified as rubber. And so these preliminary results suggest that there are a variety of microplastic sources to Lake Tahoe. And that's not surprising because there's a lot of human activities occurring in and around Lake Tahoe. And so the next steps of our study are to continue to do characterization of our filters. And we would like to to collect the data in order so we could identify microplastic particles per liter of surface water in Lake Tahoe. And so this is what we'll be focusing on the next few months for Lake Tahoe. And so to summarize, I've shown you that the combination of stainless steel filters and the peristaltic pumping method enable sample flexibility and streamline laboratory processing. These stainless steel filters are very promising. They have been shown to be applicable to micro FTIR for the detection of microplastics in samples. And so the next step will include continue to work on these Lake Tahoe surface water samples, but also to adapt these methods for other locations, like for snow in the Sierra and sediments as well. And so with that, I would say thank you, and I'd like to thank our funders.